Hello everyone, a very warm greeting to one and all. My name is Sri Gauri and I would like to welcome you all to the orientation session of OmicsLogic Spring programs on NGS Multi-Omics and Biomedical Data Science. In today's orientation session, we will introduce you to the collection of training programs we are offering, the program structure and curriculum, associated resources you get access to, and finally, the registration process. And before we begin, let me take a brief moment to introduce you to the team behind the program. We are India and US based bioinformatics and data science company, and we have had the wonderful opportunity to collaborate and partner with esteemed universities and industries, including Louisiana State University, Georgetown University, IIT Jodhpur, and much more. To meet the ever evolving demand of skilled bioinformaticians and data scientists, over the years we have created innovative training and research offerings, including online courses, training programs and workshops, research internships and fellowships, research services for faculties and scientists, as well as a dedicated online degree program. And for those of you who are joining for the first time, let me also briefly introduce you to the OmicsLogic Spring programs. Designed as a project-based learning initiative, they are an incredible lineup of online training programs designed to help students, faculties, and researchers familiarize with essential terminology, methods, and practical tools necessary for executing research in the field of bioinformatics and data science. And this spring, we are excited to bring to you programs on Introduction to Big Data Bioinformatics, Genomic Data Analysis, Transcriptomics for Biomedical Research, Metagenomic Data Analysis, and Biomedical Data Science using Python. We will explore each of these programs in depth during today's orientation session. Before that, I'll walk you through how these programs are structured. Over the years, we have developed innovative learning resources where students and scientists of all backgrounds can experience hands-on project-based learning. Here is where our interactive online learning environment called OmicsLogic Learn Portal plays a crucial role. Here, we help students learn through expert video tutorials, online courses, curated data set, a cloud platform where they can practice bioinformatic analysis. And sometimes while learning online, we often get stuck and require constant support. So to do this, we have a dedicated team of teaching assistants and technical team to monitor your learning progress and assist you wherever required. For learning and practicing analysis, we give two options. For instance, you can learn bioinformatics without any coding at all. And for those interested in opting the non-code pathway, they will be given directions to access the T-Bioinfo platform for bioinformatic processing and analysis of data. For those interested in learning to analyze data through coding, we provide the Google Colab notebooks, which will help you develop coding skills while learning popular packages in R programming and Python. And here's a quick table view summary of the resources that we just discussed. The centralized learning platform called the OmicsLogic Learn Portal is where all the asynchronous online courses and example projects are present. And to supplement your learning experience, there are lecture videos that the mentor is providing. Now, to practice the analysis, there are two options, the non-coding and coding pathway. Those who are interested in opting the non-code pathway, they will be given directions to access the AI Big Data platform. And for those who are interested in coding, they can opt for Python or R programming. And along the duration of your training program, you will receive online technical support and assistance. And at the end of the training program, you will receive the program and course completion certificates. And finally, if the student is interested, they can also proceed further ahead and complete a research project by enrolling in the research program. So now I'll take you to our centralized learning platform, OmicsLogic Learn Portal, that I covered in my previous slides to help you explore the various programs and training resources. So this is the OmicsLogic Learn platform, where once the students enrolls in the training program, they can access the online courses, expert video tutorials, and example projects. So to navigate the platform, simply head over to omicslogic.com. And if you have not created an account yet, simply head over to the blue bar on the top that says login and click on the create an account option that you see here and enter few details such as your name, email ID, password, and few other details such as your university and your level of organization, and your account can be created. Alternatively, you can also create an account using your existing social media handles. This could be your Google, Facebook, Apple account, GitHub account, or even Twitter. And for those of you who already have an account on the portal, simply click on the login now option, enter your existing email ID and password, and click on sign in. So once you sign in, the very first interface that you see is the courses page. And here on the blue bar, you will see your name. Click on that and it will redirect you to the profile settings options. So here, one of the very first tasks is to complete setting up your profile. So how to do that? Click on the profile button that you see here and enter your name. Make sure that it is in proper capitalization and there is no spelling error because this name will be printed on your certificate as well. Upload a profile image for yourself and add a brief bio that consists of your educational background and your research interests. And finally, add a link to your social media handles. So in my case, I've linked my LinkedIn account. And once this is done, click on update and your profile settings will be completed. So it should be showing 100% like this way. So now in your profile settings, you will also see several tabs, such as the activity tab, where the mentor and the teaching assistants will be constantly monitoring your learning progress. Under the courses tab, it will display the various courses that you are currently enrolled in. And under the programs tab, it will display the program that you are currently enrolled in. And you'll also see on top on the blue bar several tabs as well. So to choose a training program of your choice, hover over the programs tab that you see here and click on the training program option. So once you click on this, it will display the various training program collections that we are offering this spring. And we'll explore each of these training program in depth in the next part of today's session.
All right. A very warm good afternoon, everyone. And today I'll be taking you through the different programs that we are offering this summer, the different omics data analysis programs, what we'll be covering in each one of these programs, and what Sri Gauri was telling you about the different modes of platforms or different types of platforms that we'll be taking you through for the analysis. I'll also talk about that. And towards the end, we'll discuss how you can register yourself for the program, what are the funding options, etc., etc. All right. So this summer, we are offering Introduction to Big Data Bioinformatics, NGS Genomic Data Analysis, Transcriptomic Data Analysis, Metagenomic Data Analysis, and Biomedical Data Science using Python. I'll take up each one of these programs one by one and talk about these programs, about the topics that we'll cover in these programs, and what different types of resources you will get to learn from. So, when I talk about bioinformatics, it's very common that you might have heard a lot about these different types of data, big data that has been enormously produced. So, this program is designed to equip you with the knowledge to harness this big data that we all have been hearing about and employ effective data science tools for reproducible research and sound results. So each one of these programs is divided into three different levels. The first one is a beginner level, then there is an intermediate level, and then there is an advanced level. So whether you are a newbie in the field and you just want to get started, we have the beginner level that will set the stage for you and take you through the very fundamentals. So in this program, Introduction to Bioinformatics, in the beginner level, we'll first introduce you to molecular biology, And we'll discuss about the Human Genome Project to lay out a solid foundation in genomics. Bioinformatics is more than just data. It's actually the frontier of modern medicine, and you can analyze biomarkers and the diagnostic process. You can leverage big data from genomics to tailor clinical decisions and also drive towards personalized therapy. Our journey here begins at the intersection of clinical practice and omics data that paves the way to revolutionary diagnostics and therapeutics. We'll be discussing about different omics fields here and different resources that could be used to analyze different omics data. At the core of bioinformatics is sequencing, the process that reads the very blueprint of life. And our program offers a deep dive into the sequencing technologies that have fueled the bioinformatics revolution, that includes next generation sequencing and how these technologies are applied in real world scenarios. Our exploration includes navigating the vast repositories of biological data. We categorize databases into primary, secondary, and tertiary types, each serving a unique purpose in bioinformatics landscape. You'll gain hands-on experience with various databases and understand their role in storing and also curating biological data. First off, in the program, we'll introduce you to different omics fields. First up is genomics. Genomics provides a window into the genetic makeup of organisms. We'll explore the types of genomic variations. We'll understand the implications of single nucleotide polymorphisms and learn about the significance of copy number variation and genotypes in the context of human biology and diseases. Next, we'll talk about transcriptomics, that is the study of RNA and gene expression. And in this program, we'll guide you through the entire process from sequencing reads to the analysis pipeline, culminating in a table of gene expression. You'll learn about the tools and the algorithms that transform raw data into meaningful insights. And then we'll talk about metagenomics. Metagenomics opens up a universe of microbial diversity. Our sessions on metagenomics will cover quality control, taxonomic classification, and the interpretation of metagenomic data, where we'll be using tools such as Data2, KIME2 for denoising and dereplication of sequencing data, and dive more deep into understanding the microbial communities or the diversities within our samples. Then comes the intermediate level. So in the intermediate level, you will dive little more deep into different omics fields that we just introduced you in the beginner level. We'll also focus on, you'll get to learn about genomic variant calling, annotation. We'll also focus on the visualization aspect of bioinformatics, employing powerful tools like the integrated genomics viewer, and this will allow you to make sense of the complex data and also understand the implications of genomic variations at a more granular level. And then comes the analysis of metagenomic data. Here you will learn how you can carry out different types of analysis, how you can create different types of plots utilizing the metagenomic data. And then in transcriptomics, you will focus more on gene expression quantification, exploratory data analysis, and downstream data analysis. And this level will equip you with the skills that are needed to perform advanced analysis and interpretations in bioinformatics. Then comes the advanced level. Now, until the intermediate level, you will be well introduced to different omics fields. And in the advanced level, we focus more on the tools and techniques that you can use to analyze data, extract more insights from the omics data that you will have learned about in the intermediate level. So here you will learn or you will delve into data science for omics data analysis. You learn about machine learning techniques for unsupervised and supervised analysis. And bioinformatics is all about transforming biology into a data science. So we will discuss how to generate better data, improve data analysis and integration, and use the data insights that you get for informed decision making in biology and medicine. We'll also take you through different clustering techniques, advanced classification methods, and feature selection. And then you'll complete a capstone project towards the end of the advanced level, putting up everything that you have learned into practice. and you will showcase your skills in a comprehensive project. The programs that we conduct, the mentor-guided training programs, offers a comprehensive learning experience, and you'll have access to mentor-guided sessions. You'll also have the access to doubt-clearing sessions, asynchronous coursework, tutorials, and a vibrant bioinformatics community, of course. I'll show you what asynchronous coursework and tutorials I'm talking about towards the end of my presentation, where I'll take you to the OmicsLogic Learn portal and show you where we have all the resources. Our mentors are experts in the field, and we'll provide you guidance and support as you progress through the program. Next, I want to talk about genomic data analysis. So you all know where our genomic data comes from. You know what is DNA, how it is organized, and how it is used. I'll just quickly take you through the very basics that you might be aware of though, and then we'll dive into genomic data analysis. The first concept to get started by understanding genomics is to understand how these genomics reads are generated, how do we align them, and how do we identify these different variants that might have some functional consequences on the phenotype. All of this starts from the Human Genome Project and from the different types of sequences that you have that produce different types of sequenced data. So the year 2020 marks a 70-year mark since the discovery of the structure and the function of the DNA molecule. And it took almost 50 years from that time to sequence the first human genome. But soon after the technology developed, it evolved rapidly. And by 2015, the 1000 Genome Project brought a significant number of whole genome sequences to the research community. And in 2018, over 1 lakh whole genomes were sequenced. And today, there is an ever increasing number of genomic, transcriptomic, proteomic, and metagenomic datasets that is available that describes subcellular processes with highly detailed digital data. And so is the need to learn how to work with these different types of datasets and extract meaningful insights out of them. Now, when we talk about data, there could be two types of scenarios here. Either you come from a lab, which is generating data, so you have that access. You have the access to the data, and you can perform different types of analysis, learn how to interpret data, etc., etc. But what if you don't have an access to such a laboratory? What if you're not generating data? The question is, where is this data available that you can use for an experience or for your research work? So there are several repositories out there. You all might be aware of the National Center for Biotechnology Information. It has other databases as well associated with it. You have GEO, you have TCGA, you have Bioproject, and several other specific databases which stores specific data sets, be it transcriptomic data or be it genomic data or be it metagenomic data, whatever your research question is related to it. So the question is, how do you explore these databases? How do you extract the right data set, or how do you identify the data set for your research question? How do you extract it? How do you prepare it? And then how do you start your analysis? So in the program, we'll focus our training in the beginner level to understand these basics, how you can leverage these big data repositories and extract meaningful data, as in the data that could be useful for your research question, and then start analyzing it. Now let us talk about genomics a little bit. So the suffix "om" is used to define various fields of omics in molecular biology, and bioinformatics refers to a totality. For example, the word genome was first coined in Germany. Sorry. Yeah, so the word genome was first coined in Germany in the beginning of the 20th century. And the word genomics refers to the totality of the genomes or the studies of genomes. Genomics is an interdisciplinary field of biology that focuses on the structure, function, evolution, mapping, and editing of genomes. And a genome is an organism's complete set of DNA, including all of its genes. In contrast to genetics, which refers to the study of individual genes and their roles in inheritance, Genomics aims at the collective characterization and quantification of all of an organism's genes, their interrelations and influence on the organism. To understand how genomics is used, we need to understand the analysis approach to DNA code, specifically how we can identify these changes, or I call them variants, characterize them, and interpret their significance. And to summarize, genomic variation can be of several different types. I'll not dive more deep into it. It could be SNP or SNV, or it could be CNVs, resulting in a specific type of genotypes. So, in the beginner level, we'll dive deep into the very basics. We'll start with different types of sequencers. What type of data do we get? How do we work with this data, or how do we explore this data? And then we'll try to understand what type of variations are out there. As I discussed, we will be exploring different types of repositories and understand how to extract data. At the same time, these databases have some meaningful tools associated with it that we can use to also analyze our data and extract some insights out of it. So, which databases can we use? For example, if I talk about variant calling, if I get the variants, I can annotate them using different variant interpretations or variant annotator tools and softwares or databases as well. So, which are those databases where the variance information is stored? How do I annotate them? And how do I try to understand that if I have a specific type of variant, is it really harmful or not? The next question that I want to answer is, when we talk about sequencing data, we have these different types of data file formats that we get from a sequencer. It's very important for us to understand the different types of file formats that we get from the sequencer. What are these files? What information do they contain? And how do we really work with them in our analysis? There are three different basic file types. First one is FASTQ file that provides us with reads from the sequencer. It is just a text-based file format that stores the sequences of reads and quality scores for each letter. Then you have a FASTA file that's again a text-based format for representing DNA and protein sequences. Typically, this is the reference file for the genome. And then you have the genome transfer format file, which contains genome annotation. So, for example, the GTF file will store the start and the end of each genomic element. That could be like exon, isoform, and gene. And I prefer calling GTF files as the address book of the genome because it stores where each gene would be present on the genome. In the beginner level, you'll be well equipped with all the basics of working with the genomic data. Now, to analyze such raw data, there are many different tools for genomic data analysis that are used. These include alignment of reads and full sequences, analysis of relationships between the sequences and their evolutionary divergence, measurements of gene expression, relationship between these sequences and structures, as well as somatic and germline variant calling methods. So there are different types of analysis that is possible with the genomic data and different types of tools or bunch of packages that you can use for the analysis for these different types of analysis with the genomic data that is feasible. So we'll go through the alternative paths, that is, we'll follow a no-code path through the T-Bioinfo platform, for an example. That's a big data platform that can be used to perform all of these analysis and many other analysis as well that I'll take you through using an intuitive user-friendly interface. Now, there are some code lovers who wish to dive deep into the codes and have their own code written for their own analysis. So, we'll also go through the basics of R for genomic data analysis and understand how different packages could be used for different types of analysis. Also, there are wonderful softwares and tools readily available that could be used for these analysis. So, we'll explore them as well. Now, using the T-Bioinfo server, we will be exploring the server also and through different platform softwares and tools as well. We'll be exploring two major pipelines. The first one is the variant calling pipeline, where we'll try to understand how the reads are aligned to the reference genome and how these variants are called, which algorithms could be used, how these algorithms vary, which algorithm to use when, and how do you filter the variants that are of importance, and how do you annotate these variants. Next is sequence alignment. We'll discuss about the basics of sequence alignment. So if you have multiple sequences, you need to align them. First of all, you need to know what is sequence alignment. What are the different types of sequence alignment? Why you should even bother aligning different sequences. So there are different types of sequencing alignments that we'll talk about. We'll talk about multiple sequence alignment, and then we'll also talk about pairwise sequence alignment, how it could be performed, which tools could be used, which softwares could be used that you can install on your systems, and how you can create a phylogenetic tree to understand how the sequences of different organisms um, are similar or maybe distinct in nature as compared to each other. So in many cases, multiple sequence alignment is used to find structurally important variation. So it could also be performed with amino acid protein sequences as well as with nucleotide sequences derived from DNA itself. Now, if I talk about the no-code path, as I mentioned about the T-Bioinfo server pipelines that you can run if you're interested to perform an analysis. So when you use the specific pipeline for your analysis, you can simply use the FASTA files. We'll talk more about it in depth, and we'll also perform the practical hands-on. But just giving you an idea, we simply take the input file as FASTA file, and then we consider some important steps like pre-processing, mapping, variant calling, and annotation. The basis of this program is to understand what are the different steps involved for a specific type of analysis, for a specific result that you are aiming for. What types, what tools are used for each step, what options of tools do you have, and which tool to use when, what parameters to set for each of the algorithm and why. And then finally, when you have the results, how do you really interpret the results that you have? So we'll go through a variant calling pipeline, and then we'll go through a phylogenetic analysis pipeline as well, and then build useful phylogenetic trees. So in the intermediate level, we'll dive deep into variant calling, phylogenetic analysis, and then in the advanced level, we will learn more about GWAS, genome-wide association studies. To carry out genome-wide association studies, researchers basically use two groups of participants, people with the disease being studied and similar people without the disease. And sometimes this can also include certain variations of disease or condition that is being studied. So we learn more about GWAS. We'll also dive deep into the GWAS pipeline and look how you can explore open source repositories or maybe platforms like UCSC Genome Browser and perform a GWAS study. And we'll also explore and learn how you can use R for genomic data analysis. We'll also explore several case studies in the program itself. For each one of these different analyses that we'll discuss, we'll pick up a case study that we'll take up as an example and then learn how we can interpret results for that specific research question in hand. For variant calling, we'll discuss about the breast cancer gene TP53 in general and look at some novel variants that we were able to identify. And for phylogenetic analysis, we will look into the SARS-CoV-2 study specifically. There are also several genomic project examples that we'll be discussing about, including the TP53 gene example that I mentioned. So that was all that I wanted to discuss about genomics. As I mentioned, we will learn more about R for genomic analysis in the advanced level. Next comes transcriptomics. For the transcriptomics program, again, we have these three different levels: beginner, intermediate, and advanced. And I'll take you through each one of these levels, what we learn in each one of them. So to begin with, one of the major challenges in RNA-seq analysis is related to accurate, reproducible, and effective processing of large-scale datasets that represents gigabytes or even terabytes of data. And such large quantities prevents many to gain practical hands-on feel for RNA-seq. So the specific training program starts with the introduction to next-generation sequencing and covers the principles behind it and its applications. The participants will learn about the sources for transcriptomic data extraction and gene expression quantification techniques as well. Now, as I discussed during the genomic data analysis program orientation as well, that there are several repositories and we really need to learn how to extract data from these gold mines. So we learn about these different repositories, starting from GEO, TCGA, Firebrowse, SRA, then you have Expression Atlas, then you have ENA, then there is one more called GPR, Gene Expression Profiling Interactive Analysis, and also Human Protein Atlas. Then we'll also introduce you to exploratory data analysis techniques and the downstream data analysis through differential expression pipeline, and also talk about biological interpretation. So for RNA-seq, there are several steps involved in the workflow. We'll talk about each one of these steps and what challenges we usually face and how do we overcome them. Next comes, after differential gene expression analysis, is functional analysis through pathway annotation and through pathway annotation. The participants will get to know about a range of analytical tools to apply to their own research. And by the end of the beginner level, you'll have a good understanding of the key concepts and the techniques that could be used in transcriptomic data analysis. And now there are two paths, as I mentioned for genomics as well. For each of the omics type, you have two paths, the no-code path and the code path. The no-code path will take you through the platform, we'll take you through open source tools, softwares, resources, and there are also some tools inside the databases. We'll go through them as well. And for transcriptomic data analysis, we'll go through specifically Excel, how to perform different statistical tests using Excel. And that's a very basic fundamental skill that you should have if you're working with RNA-seq data. And then the code path will include how you can use R for RNA-seq analysis. After differential gene expression analysis, you will learn about pathway analysis, network analysis, gene set enrichment analysis, and this will be a part of the intermediate level. And you'll also learn about regression analysis. And obviously, as with each and every analysis, you have the interpretation, understanding involved as well. You'll also learn about machine learning techniques in the advanced analysis or the advanced level. The machine learning techniques for unsupervised and supervised analysis of RNA-seq data, including several different algorithms of clustering and classification methods. And you'll also go through several case studies by dealing with RNA-seq analysis that will lay off a good understanding for you and help you work with your own research question. The next program that I want to talk about is biomedical data science using Python. So why this program? The demand for skilled data analysts and scientists who can derive meaningful insights from biomedical data has never been greater. And that's where our online mentor guided training program on biomedical data science using Python comes into play. So in today's rapidly evolving world of biomedical research, the generation of data has become the norm, and every day vast amounts of valuable biomedical data is being produced that holds the potential to revolutionize our understanding of diseases and also pave the way for groundbreaking medical discoveries. However, the sheer volume and also the complexity of this data presents a significant challenge, and that's where data analysis comes in. So, this program is designed to equip you with the knowledge and the skills that you need to excel in the field of biomedical data science And through a combination of different resources that we have put together, you'll be able to unlock the potential of high throughput biomedical data. The beginner level of this program will serve as a solid foundation in Python programming and its application to biomedical data science. Now, if you enroll in Python for biomedical data science, let's say, it's not that we directly jump into omics data analysis. We'll obviously introduce you to Python basics first, and then we'll talk about different types of omics data, what it looks like, what you should be expecting in that specific omics data, and then how do you analyze the data. So you'll start by understanding the basics such as data types, variables, control structures, functions, and modules as well. And then you will dive into data processing, cleaning, using powerful libraries like NumPy and Pandas. And once you're armed with these skills, you will move on to exploratory data analysis. You will learn how to you can leverage visualization libraries like Matplotlib and Seaborn to uncover the patterns and the relationships as well that are hidden within your data. And you'll also gain an understanding of dimensionality reduction techniques and machine learning using scikit-learn. Now, as you will progress to the intermediate level, you will deepen your understanding of clustering and advanced machine learning techniques specifically tailored for biomedical data analysis. You will explore clustering techniques like k-means, edge clustering, spectral clustering, use Python clustering packages like scikit and scipy, and you'll also learn model validation techniques and evaluation matrix for supervised machine learning models, and you'll delve into advanced machine learning techniques as well, like decision trees, random forest, SVM, LDA, and UMAP, and apply them to real world biomedical data sets. Now, we understand that you are a biologist and you might be feeling stuck getting started with coding languages. That's why we have put this program for you to have it simplified for you, and we'll take you through the paths on the portal shortly. So on the portal, I'll take you through how this program has been put together, where you will learn the code block and then you will practice it from visualization of different omics data to exploratory analysis and hypothesis testing. We also have put together Google Colabs for you to practice through for both the languages. Now, you might be confused which specific program to pick. So, if you are confused, you can maybe discuss with us your specific requirements or your specific interests, and then we can show you a roadmap that you can maybe consider and get started with your learning experience. Last but not the least, I want to talk about the field metagenomics. Some of you might be aware of what this field is all about, whereas some of you might be new to this field. So I'll not go much deep into the sequencing types right now, but in the program, we have divided it into three different levels: beginner, intermediate, advanced again. In the beginner level, we talk about different types of metagenomic sequencing techniques, how this data is produced, and the very basics of the sequencing and the type of data that you would be having. Now, why do you even why like this program is important for the people who are in the field of microbiology? So we know that indigenous human microbial communities really contribute to critical metabolic, physiological, and also neurological functions that includes differentiation and maturation of host mucosa and immune system, or food digestion and nutrition, or energy generation, or metabolic regulation and control of fat storage, processing and detoxification of environmental chemicals, also maintenance of skin and mucosa barrier function, or development and regulation of immune system and its reaction patterns, and also prevention of invasion and growth of disease promoting microorganisms. These are just some functions of microbiomes. There are a lot more interesting functions where microbiome plays a crucial role in understanding what the microbiome is composed of, and how the dysbiosis in the microbial community can really lead to very harmful conditions, and how we can overcome them is the key for this program so that you understand how the data can be analyzed using different tools and resources, and then come up with your own research question to mitigate the problem. Now, in metagenomics, there are some specific types of analysis that you basically look at. Uh, I want to talk about taxonomic classification here that plays a crucial role in understanding the microbial composition and the diversity within a complex mixture of organisms. And one commonly used approach for taxonomic classification is the utilization of the small subunit ribosomal RNA gene, 16S RNA gene. Why this gene is specifically used, why do we focus on that? We'll dive more deep into it to understand it in the program. Now, once you have the data, the next step is that after the data has been analyzed, you have the sequences. Now, once you have the read sequence from the sequencer, you want to map them onto the reference databases. So there are several different databases that you will learn about how you can use the sequences on these databases as a reference genome or as a reference sequence, and then try to understand what normal sequences you might have for which a reference genome might be missing. Now, all of this seems easy when you start discussing the process or the flow, but we have dedicated pipelines for this type of analysis. The two pipelines that I mentioned about in the beginning as well, the Data2 and the Kind2 pipeline, we'll be going through these different types of pipelines, what these pipelines do, and why do we have these so many steps, and what parameters are we taking. We'll dive more deep into it in the program. So these two pipelines basically help us with the taxonomic classification of the microbial samples that we take for a study, and helps us understand who is there in the sample, or what type of communities are there in the sample, and in what or how much abundance are they present in the sample, and what is the normal abundance and what is an abnormal one, and what this abnormal abundance could really lead to. So that would be the aim of the study here. And then we also have the phylogenetic trees that we'll get for the microbial sequences that we'll be dealing with. And we'll also generate some beautiful plots to understand the differences within the microbial samples that we have, or between the microbial samples that we have, and also draw some abundance bar plots. Not only this, we'll also go through some very interesting metagenomics projects that have already taken place, and also learn what the outcomes were of these projects. So we'll discuss about the American Gut Project. We'll also discuss about gut microbiome and its role, gut microbiome and its role in healthy gut function, in chronic diseases, in cancer, and also we'll try to understand the response to treatment. And most importantly, we'll also discuss about the gut-brain axis. So that was all that I wanted to talk about for these different types of omics data analysis programs that we have for you this summer. And now I'll like to take you through the OmicsLogic Learn portal and give you an overview of different types of resources that you'll have for the program. Meanwhile, if anyone has any question, please feel free to raise your hand or put your question in the chat. Otherwise, towards the end of this session, we'll have the stage open for all of you to unmute yourself and have this discussion. Okay, now for those who are new to the OmicsLogic Learn platform, I'll share the link in the chat. Please create a login for yourself. Once you are on this link, you can see the home page here. You just need to click on register for free and then create an account. Creating an account is absolutely free. So once you have created an account, you'll be able to see your profile like this. I've already logged in. I'll take you through my profile in some time. Before that, I want to take you through the courses, and then I'll show you how my course progress could be monitored on my profile, and where do I get the certificates. Okay, so For this program, you have to keep these two tabs online. For the programs, if you click on training program, you will get the list of training programs that I took you through today. The first one is biomedical data science using Python. You have introduction to bioinformatics, transcriptomics, genomics, and I think metagenomics is on the second page. All right. And I'll also discuss about the research program. Yes. All right. Now, for each one of these programs, we call them training program pages. Once you enroll in the program, you will get the access to this training program page where all the resources are put together. So on the training program page, you have these different levels like this: beginner, intermediate, advanced. Now, if I click on one of these levels, I can see that each level has got 6 sessions. Beginner level has 6 sessions, and the intermediate one also has 6 sessions, and so does the advanced one. Now, for each session in each level, you have these resources put together here. So you can see a session video here for the first session, and you'll need to go through it. These videos are unlisted videos of our channel that are put together on the training program, and only the program participants can access these videos. Now, once you go through the session videos, after that, you can see that you have some resources linked here. The first resource says associated lesson, and then you have additional reading material. Reading material could be research papers, could be articles, or maybe it could be some videos from other sources that we have put together here for you, just for you to go through it if it's important. And also you will have some virtual lab activities put here as a resource if it's important for that session. And also you will find that in some of these sessions where we perform a practical, you'll have the link to the demo pipeline of the server. On the server, you have these demo sections or the example sections that you can go through and run the pipeline and see what type of results you get and how the results are to be interpreted. And also you will find the link to the data set that has been used for the practical hands-on. And also you will find the link to the database or the repository that we are using in that specific session, in the practical sessions only. Now, I was talking about the associated lesson. So for every training program, you get the access to different courses. These are the courses that are present in this course tab. So with this training program, you get the access to these different courses here, okay? And for each one of these courses, you have different lessons that are associated here. I'll show you what is this. So I'll open up one course, Introduction to Bioinformatics. I'll open it in a new tab, and I'll also open this associated lesson. Now, if I go to the course here, you can see that each course here has different lessons. And it's just like a book where you have these different chapters, and each chapter has the concept put together in the form of textual format or in the form of images or in the form of videos, lectures. And towards the end, you also have some quizzes and assignments that you have to complete in order to complete the lesson. All right. Now, what happens is this is the course that I opened from here, but we have added an associated lesson for each session so that you just click on that lesson link and you know that this is the specific lesson that you have to complete for that session. It's that simple. So as I mentioned, in Introduction to Bioinformatics, we talk about different omics courses. Genomics, metagenomics, transcriptomics, and single cell as well. We're just introducing these courses to you. We will not overwhelm you with lots of concepts or with a detailed advanced level of understanding, I'll say. But yes, we'll introduce you to these different concepts, and we'll also introduce you to the programming languages R and Python. So that was about a training program page and what it includes. All right. Other training program pages are quite similar. It's just that the content is different. Obviously, it has to be because the topics of each program are different. Now, on my profile, you can see some points and you can see these badges. What are these? For each course that you complete or for each step in the lesson that you complete, you get some points, and these points appear here. And now coming to the badges. So I'm not going through each training program page right now because it's the same. You have the same resources, the same fashion of content that you have to follow. And you can take your time, go through these training program pages, go through the sessions that are out there so that you get to know what topics would be covered in detail, and then sign up for the one that is the best for you. And if you're confused, you can reach out to us anytime. Now, if I go to my profile, I want to answer here two things. Where do you get your certificates? And how do you get to the training program? The first option is you go to the programs, training program, pick the program that you enroll for. The best way is you open your profile and you can see the badges here. So you simply click on the badge of the program that you have enrolled for, and if you click on it, you will get redirected to that specific training program, and then you can go through whatever session you are going through as of that moment. And this specific dashboard helps us monitor your activity, and also you will get to know how many points you earn per step that you complete. Now, once you complete a specific training program, after you complete this session, let's say I went through this session, need to click on complete. I've completed this already, but if you have not completed, it will say complete. So you click on complete and then you go to the next session like that. Once you complete all the sessions of your training program, you will get your certificate under your profile. So you click on this certificate and then you can simply download it. Here is the download option, right? So that is how you can download your certificates. Now comes the courses tab. So let's say you want to explore what all courses are there and you want to learn a specific coursework. So you simply go to the courses tab and you enter a keyword. For example, I'll write metagenomics. And then you get metagenomics, hit, and then you go through it. Now you can see there are several projects as well associated with this specific keyword metagenomics. These projects we have put together as case studies for you to understand that. So this specific coursework will first of all tell you what types of tools are out there, what is the workflow of analysis, what tools to use, how to interpret your results, etc., etc., using the platform or open source tools or softwares, or maybe R code, right? But an example project will help you understand, once you learn the skill set, how do you answer a research question in hand. So there are different research questions that we have tried to answer through different case studies on the portal, and you can explore them to understand what approach was used and how. I want to show you one more thing, uh, that is Python. So for Python, you have two courses. The first one is an introductory coursework, and the second one is a sort of introductory coursework, but takes you to an intermediate and then an advanced level. So here we get you started from the very basics. Even if you don't have a background in Python, you can get started with this course. Then you have the lessons given here for how to import data, how to visualize data, how to look at the outliers, and then perform descriptive statistics, perform different dimensionality reduction methods to reduce the dimensions. Then you have some lessons on machine learning and deep learning as well. And yeah, that's it. Okay, so now if you click on a specific lesson, I want to show you what's inside there. So for the programming sessions or the lessons, you have the content put together in this format where you have the syntax of the code syntax given first of all, and then the explanation for the same. All right, and then the explanation for the same. And towards, this is a pretty lengthy lesson. I want to show you the collab. So you have the output here, what type of plot you will get, and all the different packages have been explained. The code block has been explained. So you can practice it from here. I want to show you a collab. This is a pretty lengthy lesson. So I'll open a short lesson maybe. I'll just scroll down. Okay, I'm completing these steps just because I want to reach the practice code. It's better you don't do that. Just complete the steps when you actually complete it. So here there's a Google Colab. Okay, now if I simply click on it, you will be redirected to a Google Colab. Now, in this Google Colab, this collab is for your practice. So this collab has all the code blocks that we discussed in a specific lesson. So what you are supposed to do is you have to click on File, then you click on Save a Copy in Drive. It will save a copy in your drive, and then you practice the code block and do your stuff. Otherwise, what you can do is open a new collab in your drive, and then you type the code blocks with an understanding and then practice it. That is also a better way to work with the collab. For those who are new, for those who are working with Python, the default environment of collab is Python, so you're good to go with it. But for those who are interested in learning R, on the platform there are also courses on R for biomedical data analysis. You can change the runtime type to R and then use the same collab to also write R codes, just for those who are new to collab. So that is how you can practice codes. Now you can definitely practice codes on Jupyter Notebooks or RStudio as well, but we prefer collabs because if you encounter an error, you can simply share your collab with us and it will be easy for us to look at your error and resolve it with you over an email or a Zoom call maybe. So it's easy to track the errors and then resolve them. That is it. That was what I wanted to share with all of you. Okay, so now comes the research program. So what is the research program all about? Once you have gained the specific skill set of a specific type of omics field, you know what type of analysis has to be done and what type of data you can use. The next step is you can join a research program. In a research program, it's little different from the training program. In the research program, you work closely with the mentor on a research question. Now, this research question can either be proposed by you, or if you're interested to, let's say, work on transcriptomic data analysis or transcriptomic based research project, but you don't have a research question, you can reach out to us. We have already put together some research questions for research fellows to work on. We can have a discussion, we can show you these research questions, and you can pick one. Now, for different omics fields, there are different research specialization tracks that you can pick to carry out your research work. You can either work in the field of oncology or infectious diseases or maybe metabolic disorders or agriculture or space omics or precision medicine. So there are different research specialization tracks that you can pick and have a research question prepared, and then pick up a specific type of omics field and then work on your research project. Now, how does the research program work? Either you can enroll for 2 months, 3 months, or 6 months. Here you have these different levels that you can enroll for. Sri Gauri will explain you further how you can register and how you can change the currency and what are the scholarship options. But I'll just tell you what it includes. So in the 2 months of the research fellowship program, it's more feasible for those students, be it bachelor students or master students, who want to work on a research project either for an experience or maybe for putting up an application for higher studies or maybe preparing a poster for an exhibition or a science fair or for putting up an application for a master's program or higher studies maybe, and best suitable for the ones who want to work on a college or a university dissertation. All right. in 2 months, we would be expecting that you are already trained on a specific type of analysis, and then 2 months you will just focus on research project. Now, in a training program, we only meet when you have a question or when you encounter a question. But for a research program, the mentor will be having a meeting with you via Zoom, and will discuss your path. Will discuss what steps you have to follow next. Will discuss your problems where you are stuck. Will resolve them, and then will tell you the next steps in your research. So that way, you discuss with your mentor how you have to proceed. Now, it's not that you just stay in touch with your mentor through these Zoom meetings. We also have Skype groups created for these different research fellows. So, when you are going through a specific step of your analysis and you're stuck somewhere, you can reach out to your mentor for assistance. Now, for 2 months, I mentioned when how 2 months of research work can help you and what type of outcome you can expect from a 3 months of research project. It's just that as the time increases, it's just that you can shape up your research in a more smoother way, in a better way, and move it towards a publication level sort of research. So a 3 months of research is best suitable for maybe PhD scholars or students as well, BSc master students who want to get an exposure with a specific omics analysis and have a research publication out of it. And many students come to us and ask us how much of work or how much uh, time they should put for a specific type of outcome in 2 or 3 months. It really depends on you. If you are a PhD scholar, we might not expect you giving 3 to 4 hours every day because you'll have your own work as well. Or if you are working on a project that is related to your own PhD work, then you might be giving more number of hours. Or if you are a college student, you will have your own classes as well, and in the evening you're working on a project. Or if you're working on a dissertation, you will have more number of hours. So this question of how many number of hours you should dedicate to a research project is it's a very different question for everyone. So I prefer that if you're interested in a research program, you first of all schedule a meeting with us where we can discuss your availability, what type of experience you want to have, what type of data you want to work with, what type of research question you are interested to work on, and then we take your application further. Now, a 6 months of research project is suitable for students who want to have a research publication in a good journal with a good study, and then they take this the 6 months of program where they work on it extensively. So that is how these different levels vary. And if there are any questions, please feel free to ask in the chat, or maybe you can unmute yourself as well.
Now, here's a quick glimpse into the various ways that students have leveraged these resources to work on different research specialization track. So, for instance, our Python training program was part of the Systems Medicine graduate program conducted at the Georgetown University. And the training ended with students submitting their research project on the OmicsLogic Learn portal. And as you can see, students have developed machine learning models using Python to address research questions in various disciplines, including infectious diseases, metabolic disorders, neuroscience, oncology, and so on. And further, students also have the opportunity to participate in poster presentation competitions and win exciting cash prizes. And twice a year, we conduct Omics Research Symposium, wherein we invite experts from all around the world and provide their feedback for the students' research work. Now, there are also examples of student research publications, and here is one example of Prithvi, who was an undergraduate student, who conducted transcriptomic analysis on dataset from multiple sclerosis patients and published her research in an open access journal on immunogenetics. Simay Dolaner is another student who went on to complete an oncology based research project on leukemia and published her research in Columbia Undergraduate Research Journal. And the student projects are not just limited to graduates and doctorates. For instance, Andre was a high school student when he participated in the research program, and he completed a neuroscience project that he presented at the Regeneron International Science and Engineering Fair, for which he won the Potemkin Prize. And these projects can also be showcased for higher studies and university applications, like some of our research students have done, such as Dilara Daikin, who got accepted to the Erasmus IMBRC program in marine biology, Dhruv Mehra, who got accepted to the precision medicine graduate program at University of Manchester, and Anusha, who got accepted to the PhD program. And faculties and researchers have also collaborated through our programs to publish their research work. And here's one of the latest collaboration that we conducted with a team of researchers at Nebuchi Memorial Institute for Medical Research at the University of Ghana. And it is also possible to pursue an entrepreneurship and business innovation track, like a team of industrial researchers from a biotechnology company in Nigeria did. So they developed a microbiome test kit to improve the fertility of women in Nigeria. And such tracks are pursued not only by researchers, but by undergraduate and postgraduate students as well. For instance, this was a group of students who worked on developing an early detection kit for Alzheimer's disease using metagenomic datasets. And now we consistently share these updates on ongoing research project outcomes, student publications, exciting job opportunities, and latest article on our social media platform. We'll share the link to our LinkedIn group where you can explore all of these updates. All right, so as we discussed, one of the very first interface that you see when you log in is the courses page. To view the various training programs that are being offered, simply head over to the Programs tab on the top and click on the Training Program option, and it will display the set of training programs that are available. So here, when you scroll down below, you will see a search bar that says looking for a specific program. Here you can enter the keyword that you're interested to learn more about, or you can explore each of the training programs one by clicking on the next page. But here, let's take the example of Python since it's popular. When you click on the program, it will redirect you to the program page where all the necessary information are available to the participant. So here you can review the session topics that are being covered within the program and various levels that are available within the program as well. And as you scroll down below, you will see the pricing options for each of the training levels as well. You can change the pricing option by clicking here. So for example, right now it is in INR. I can change it by clicking here in the USD as well. So do make sure to review the training program level options that are there and the associated resources that are available. So each of the training level have different varying set of duration and the associated resources. Their access will also be varying depending on the training level that you're choosing to enroll. So you can review these details here on the program page. Whenever you click on it, all the program page have the same set of training levels, so you can review it once. So for the beginner level, it's $100. For intermediate level, it's $200, and for advanced level, it is $300. So in the next step, you can just click on the buy now option in the training level that you're interested to purchase. So here I'm doing it for the beginner level, and enter your key details such as the user details and so on, and select the payment option such as PayPal or Razorpay, and the program can be purchased in this manner. So that was all that I wanted to cover in this part of the session. So if the participants have any queries, you can reach out to us on communication@omicslogic.com. And if the participant is interested in opting for the training plus research option that was covered, you can also email us about that, the duration about the research program you're interested to enroll, and we can suggest you the flexible training option as well as the pricing. Thank you so much.